Hi, I'd like to welcome you to our first video for the first part of lecture 10, and we're going to talk about photoreceptors. And as you guys are all used to, photosynthesis is used to produce energy. The plant absorbs light, and that light is converted into sugar. And so we all know that light is really important, obviously, the source of energy for photosynthesis. But light is also recognized by plants as a signal for developmental processes. Light will stimulate germination, photomorphogenesis, photonasty, and flowering. So here, right here, is a picture of photomorphogenesis. We'll be talking about this in detail. You can see that seedlings, when they are grown in the light, they're shorter, the little leaves are green. When they're grown in the dark, they're super long. They're kind of, in a sense, they're searching for light, and so they grow super long and they don't produce chlorophyll. Here is a picture of photonasty, and you can see that at different parts of the day, the leaves are kind of upright, so they can receive more light. While at other times during the night, the leaves kind of move down. So this movement of the leaves in response to light is called photonasty. Later, we'll talk about flowering, and we'll see that flowering is also regulated by light. And so just quickly want to kind of talk about the details of photomorphogenesis and skotomorphogenesis, which is growth in the dark. These traits are used a lot in research to understand photoreceptor function. So a plant that germinates in the light will have a short hypocotyl. And the hypocotyl is the embryonic stem. And a plant germinated in the dark, the hypocotyl elongates. A plant grown in the light, the embryonic leaves are cotyledons. So here we are looking at a dicot. There are two cotyledons. We can see that they are kind of open. They have a wide angle between them. And they would be green because in the light, chlorophyll biosynthesis is stimulated. On the other hand, in the dark, if you're following skotomorphogenesis, the apex is still kind of hooked down. So we call this, the apical hook is closed. And we also see the cotyledons are really close together. Okay, so these guys are close together. Here, the apical hook is closed. And as we saw in the last picture, in the dark, you can't produce chlorophyll, so the cotyledons here are yellow. And they are yellow because you can produce carotenoids in the dark. They're another type of pigment, but you can't produce chlorophyll. So here they're green because you can produce chlorophyll. Okay. All right, so that's the difference of photomorphogenesis in the light and skotomorphogenesis in the dark. Now, a number of photoreceptors have been isolated, and I just want to introduce you to those. Today's lecture, we're just talking about phytochromes. And here is the visible spectrum, and you can see it goes from, well, part of it is visible, part of it is UVB and UVA light. But we're going to be talking about far red light, and we'll also talk about blue and UVB. Okay, so phytochromes, they absorb red and far red light. Cryptochromes in phototropins, they absorb and respond to blue light. And UVR8 is a UVB responsive promoter. Photoreceptor. But again, today we're focusing on phytochromes. For convention, here's just some of the terms we use. Phi A similar, lowercase italics is the mutant. Phi A uppercase italics is the name of the gene or the messenger RNA. phi A, uppercase no italics, that would be the apoprotein. And the holoprotein, which is the protein plus the chromophore, that's kind of the new term here. This is listed as lowercase, but not italics. So these are kind of the rules of the field. We're going to see that photoreceptors work together for many responses, and we're going to be looking at in a few slides action spectra. And you basically expose a plant to many different wavelengths, and you measure the response. And from here, you can often determine which photoreceptors are mediating a particular response. Okay, so the first kind of discovery of the phytochrome photoreceptor were studies done with lettuce seeds. And it turns out lettuce seeds, they need light to germinate. So people in the past, they knew that if you put lettuce seeds in the dark, they wouldn't germinate. And when they tested different lights, and we'll look at those data in a moment, they saw that when they exposed the seedlings to red light, and just not even a long, maybe a few minutes of red light, that would stimulate germination. You can see the little radicals emerging from the little lettuce seedlings. If the red light was then followed by far red light, again, just for a few minutes, then this would reverse the stimulation of germination. So red stimulated, if the red was followed by far red, it reversed it. And again, if you had the red, the far red, and red, then it stimulated. And again, if you continued this red, far red, far red, then inhibited. So it seemed that the photoreceptor could be in an active or inactive state. And now we understand this, as we call this PR is the inactive form. And this absorbs red light, and it photoconverts into the active PFR form. The active form here is going to stimulate germination. Okay, the active form PFR, it can absorb far red light, and it will convert into the PR, or the inactive form. So right here is kind of our basic and foundational understanding of phytochrome, and we're going to learning more about the details, molecular details of how this whole thing works. But this is kind of the key understanding here. Okay. And so here are some action spectra. And again, in these experiments, they took the lettuce seeds, and they exposed them to different wavelengths of light, and they measured the germination response. And you can see here that the stimulation of germination, it peaked right here. Sorry, this is red. And it peaked right about this wavelength, okay? And when they looked at the inhibition, they found that this peaked right around here, okay? And this is in the red, and this is the far red. And what they noticed, so they have these action spectra for quite some time. And later when people started studying phytochrome, they were the first place that was purified from were oat seedlings. And they purified oat phytochrome, because you can just get a lot of seeds, like, kilograms of seeds, and purify the phytochrome. And when they did this, they saw that PR, which is the one that absorbs red light and gets converted to the active form, its action spectrum, its peak, almost coincided exactly with the stimulation of germination. And then here they have a PFR. They took the oat phytochrome and put it in the PFR form and tried to see where it absorbed light. Its peak was 730 nanometers, very close to the inhibition of germination. And so, so this is what we call the absorption spectrum of the purified photoreceptor, and we're comparing it to the action spectrum for germination. We can see that they match pretty nicely. Just a note here, we're going to talk about shade avoidance probably actually in the next lecture, but I just want to show you here that in this graph, they show you light filtered through shade, like if you have a leafy canopy and the light filters through the leaves, it's really enriched in far red light. And when we talk about shade, you'll see why this is important. Okay. Here is another action spectrum, and this is for a response that's not mediated by phytochrome. And here, this is phototropism. And if you guys remember, when a seedling germinates, if it is exposed to light, it will actually bend towards that light. Okay, so that is phototropism. And when they looked at an action spectrum, instead of just exposing it to sunlight or white light, looking at the different wavelengths of light, they found here in this experiment, they're looking at purple, blue, and green. They found the action spectrum, the peak of it was about 440, and that was really close to the peak for phototropin 1. And we actually talked about this in the paper that we talked about where they had that optogenic blue light receptor, and that was just the LOVE2 domain absorbing FMN. But phototropin is the actual plant blue light photoreceptor. Anyway, so this is an action spectrum for blue light phototropin, which we'll be talking about in the next lecture. Okay, so here what we're going to be doing in these online lectures is we're going to stop and we're going to pause to answer some questions. There is a Word file that has all these questions. You can use that to write your answers. As I mentioned in the Beachboard message, you will not be having writing assignments while we're doing this online lectures, because you'll see there's quite a few questions that are associated with lecture. But now what I expect is that when you write your answers to these questions, that you use really good English, and it's a good way to practice your writing skills. The first one's super easy. It actually doesn't have to do with writing skills. You're just going to fill out the table, name the part of the seedling 1 and 2, and indicate the phenotype in the dark or the light. And I'm kind of giving you what I'm looking for here. Question number 2, I'm asking about the lettuce seed germination experiment. And then question 3, we're talking about action spectra. Okay. All right, so go ahead and this is the first part 1 of this lecture.